lytic cycle in T11 bacteriophages. The multiplication cycle of bacteriophages, like that of all viruses, is completed in 5 distinct stages. First, attachment or adsorption. Second, penetration. Third, biosynthesis. Fourth, maturation. And fifth is release. Attachment. As a result of chance collision between phage particles and bacteria, attachment or adsorption occurs. The tip of the virus tail becomes attached to the cell via specific receptor sites on the cell surface. T11 bacteriophages use tail fibers as attachment sites. The specific receptor sites of the bacterium is part of lipopolysaccharide. Although any surface structure can function as specific phage receptor, including flagella, pili, carbohydrates, and proteins in membrane and cell wall. Penetration. The actual penetration of phage into the host cell is mechanical, but it may be facilitated by localized digestion of certain cell surface structures, either by phage enzymes, for example, lysozyme carried on the tail of the phage, or by viral activation of host degradative enzymes. During the process of penetration, the tail sheath of the phage contracts and the tail core is driven through the cell wall and the plasma membrane. When the tip of the core reaches the plasma membrane, the DNA from the bacteriophage's head passes through the tail core, through the plasma membrane, and enters the bacterial cell. Therefore, the phage particle injects its DNA into the bacterial cell the way a hypodermic syringe injects a vaccine. In biosynthesis, the sequence of transcription events in the viruses may vary from each other, but the general mechanism is the same. The biosynthetic mechanism of phage T4 is discussed here as an example, which occurs in several stages leading to the formation of immediate early, delayed early, and late gene products, so named on the basis of sequence of their appearance. Initially, the phage uses the host cell's nucleotides and several of its enzymes to synthesize many copies of the phage DNA. Virion-specific proteins appear later, followed by appearance of organized capsid precursors and resulting in the formation of mature infectious capsids. The host cell's ribosomes, enzymes, and amino acids are used for translation. Genetic controls regulate when different regions of phage DNA are transcribed into messenger RNA during the multiplication cycle. For example, immediate early phage genes code for nucleases that break down host DNA and for enzymes that alter the bacterial RNA polymerase, so that it will be preferentially transcribe delayed early phage genes. Delayed early phage genes code for phage enzymes which are responsible for formation of unique phage DNA constituents. Delayed genes also code for polymerases and ligases that play specific role in phage DNA replication and for RNA polymerases that transcribe late genes. Late gene products include the structural components of new phage particles, for example, head, tail, and tail fibers. They also include a phage lysozyme, which is an endolysine, which will lyse the bacterial cell, releasing the mature virions. Maturation. In this process, the bacteriophage DNA and capsids are assembled into complete virions. The phage head, tail, and tail fibers are assembled separately following 3 different pathways, each being genetically controlled. There are about 50 architectural genes which control the synthesis and assembly of new viruses. Near about 20 to 22 genes control synthesis of basal palate, tube sheath and tail. About 18 to 20 genes control the synthesis of head, while as 8 to 10 genes are responsible for the synthesis of tail fibers. Soon the head is filled with the phage DNA and attached to the tail spontaneously and is followed by the assembly of tail fibers. The last stage, release. This is the last stage of virus multiplication. The term lysis is generally used for this stage because the plasma membrane actually breaks open. Lysozyme, that is endolysine, which is coded by late gene products, causes the bacterial cell wall to break down and the newly bacteriophages are released from the host. One step growth cycle or multiplication cycle. The time that elapses from phage attachment to the release of progeny virions is termed as viral multiplication or replication cycle. Various stages involved in the multiplication of the phages can be demonstrated experimentally in what is called as one-step growth cycle. During the first 10 minutes or so after injection of phage DNA into the host cell, no phage can be recovered by disrupting the infected bacterium. This is termed the eclipse period. At the end of this period, mature phages begin to accumulate intracellularly until they are released by the cell lysis. The time from infection until lysis is the latent period. The extracellular phage number increases until it reaches at the end of the multiplication cycle. This time interval is termed as the rise period. The yield of phage per bacterium is called the burst size. The lysogenic cycle. Lysogeny is a transforming interaction which results in the integration of the viral genome into the host genome and the permanent transformation or alteration of the host cell with respect to morphology, growth, habit and the number. In lysogeny, the phage remains latent or inactive. The viral DNA of the temperate phage, instead of taking over the function of cell's genes, is incorporated into the host DNA and becomes a prophage in the bacterial chromosome, thus acting as a gene. The bacterium metabolizes and reproduces normally and the viral DNA is transmitted to daughter generations. However, sometimes the viral DNA is removed from the host DNA and the lytic cycle occurs. This process is called spontaneous induction. Mechanism of lysogeny. The lysogenic cycle has been well studied in the bacteriophage lambda, which is generally considered to be a typical of the temperate phages. The penetration of the bacteriophage is similar as in case of lytic cycle. The linear phage DNA becomes circular and the circle can recombine with the circular bacterial DNA. The inserted phage DNA is now called a prophage. Most of the prophage genes are repressed by two repressor proteins, the immunity repressor and the CRO repressor. These repressors stop transcription of all other phage genes by binding with operators and thus preventing the formation and release of new virions. The bacterium metabolizes and reproduces normally and the viral DNA is transmitted to daughter generations. Lysogeny results into permanent transformation or alteration of the host cell. Three important results of lysogeny are: first, the lysogenic cells, that is host cells, are immune to reinfection by the same phage but susceptible to infection by other phages. Second, it makes specialized transduction possible. Third, lysogenic conversion. That is, the host cell may exhibit new properties, for example, Corynebacterium, diphtheria, Streptococci, Clostridium botulinum, and Vibrio cholerae can produce toxins only when they carry prophages. In this type of transduction, once the prophage gets excised from the host chromosome, it carries genes from the bacterial chromosome and transfers them to another bacterium. After infection, as has been seen in the bacteriophage lambda, which picks up GAL genes for galactose fermentation from galactose positive host to galactose negative cells. Thereby transforming them into galactose positive. Lysogeny has also been seen in certain animal viruses such as herpes simplex virus, cancer causing viruses, and to some extent autoimmune deficiency viruses follow same mechanism.